Welcome back. In the previous session, we have discussed about Mendel's monohybrid cross, where Mendel he selected pure breeding homozygous tall plant, crossed it with the pure breeding homozygous dwarf plant, and in the F1 generation, he obtains all plants that are tall, and then he selfs them F1 into F1. In the F2 generation, he obtains 75% tall and 25% dwarf. Based on this, Mendel proposes the two laws, law of dominance and law of segregation. Today, we will discuss about three important topics. One is the dihybrid cross, incomplete dominance and codominance. So, let us get started. At the end of the monohybrid cross, there is a question. For example, how to know what is the genotype of the F1 plant? For example, when capital T is crossed with small t, now because we are using the alphabets etc., we know that this tall plant is not homozygous, it is heterozygous, capital T, small t. But in real life, practically in the field, the heterozygous tall and homozygous tall both appear to be exactly of the same height. How do you know whether it is a pure homozygous plant or a heterozygous plant? For this, we have what is called as a test cross. As the name suggests, this cross is used to test the genotype of the F1. So it's a type of cross which tells us the genotype of the F1 generation. We cross the F1 generation, that is in this case capital T, small t, with the recessive parent. So test cross is a cross done between the F1 generation and the recessive parent. Recessive parent in this case is the dwarf plant, that is small t. And when we do this, we know according to the law of segregation, during this is a parent gamete formation, alleles will separate into different gametes. So here there are two alleles. So one gamete will receive the dominant allele, another gamete will receive the recessive allele. In this case, both alleles are same. So all the gametes will receive only one type of allele. So the result will be, we get a tall plant and a dwarf plant. So the ratio of tall and dwarf plant is 1:1. If after crossing a F1 plant with the recessive parent, if you get 50% tall and 50% dwarf plants, we can say that the F1 is a heterozygous plant. So test cross is done to check the genotype of the F1 plant. Of course, if it is homozygous, if the F1 is capital T, that is both are dominant, what result will you get? You will get again the same result. The next type of cross, that is dihybrid cross. This is a cross that contains two pairs of contrasting characters, wherein Mendel has chosen a pure round plant with your yellow seeds. Round and yellow are dominant, and he has crossed this with another plant which is also pure, but it has got wrinkled and green seeds. So that is the parent, round yellow plants and wrinkled green plants. You can use any combination that you want. You can take round green and wrinkled yellow as well. Whatever trait that you take, the result will be exactly the same. So it is a cross that has two pairs of contrasting characteristics, round and yellow. These are the two pairs, and wrinkled and green. As you can see very clearly, this is dominant as represented by the capital letter, whereas this is a recessive trait. Now, this is your parental generation. Let us mark it as P. Now, the next step is gamete formation. We know that during gamete formation, alleles will separate. Now, this is where you have to pay attention. In the monohybrid cross, it is relatively simple because there is only one pair of characters. Now, here there are two pairs of characters, which means that during the gamete formation, when these alleles separate, both the round allele and the yellow allele will go together into one gamete. So the gamete will contain round and yellow because both the pairs are present in the same plant, isn't it? So during gamete separation, the alleles will also get segregated like this, round and yellow, and of course another gamete will also have round and yellow. So I am only representing one gamete. In this case also, the allele will contain wrinkled and green. So this is the gamete. This is a diploid set. It contains the full set of chromosomes. Gametes are haploid. They will contain half the number of chromosomes. So half from this and half from this. Alleles get segregated. Now, when fertilization takes place, all the alleles will once again come together in a zygote, which is now diploid. Diploid zygote will contain all the alleles. So the F1 generation will have all the alleles. This is our F1, and it was observed by Mendel that the F1 plants were all round and yellow. Law of dominance, because we know that in a hybrid condition, in a heterozygous condition, the dominant allele will suppress the recessive allele. So in this condition, the round trait is dominant and here the yellow trait is dominant. So F1 generation, only the dominant traits will be seen. So all F1 plants are round yellow. This is, till now it is similar to the monohybrid cross. Then once again Mendel wanted to know what happens if you cross F1 with F1. So the next step is selfing of F1 into F1, self-pollination or selfing. All these are exactly same to monohybrid cross. Only thing is that there is one additional pair of trait. That is it. F1 into F1. So F1 is RRYY into RRYY. Now this will produce the gametes. That is what we will represent in the form of Punnet's square, which is the graphical representation of all the possible genotypes. So like we did here, during gamete formation, the opposite alleles will go together. So in this case, it will produce RY. This is one gamete. It could also produce capital R, small y, because both are opposite traits. So this is one combination, this is one combination. Similarly, this could produce small r, capital Y, and small r, small y. So 4 possible gametes are possible. Similarly, here also we will get the same. So ry, and small r, small y. These gametes, like we did in the monohybrid cross, we use the Punnett's square. All you have to do is take one gamete, fuse it with all the 4 possible gametes. So when fusion takes place, both the alleles will come together. So first we will start with here and cross it with this. So we get capital R, capital R, capital Y, capital Y. So if you see, that is nothing but your round and yellow. Round yellow. Next, this we fuse it with this, that is capital R, capital R, capital Y, capital small y. So it is round and yellow, because yellow is dominant over green. So this is also round and yellow. Then this into this, that is capital R, small r, capital Y, capital Y. It is also round and yellow, and capital R, small r, capital Y, small y. It is also round and yellow. Similarly, we do this for this with this, capital R, small y, capital Y. That is also round and yellow. This and this, capital R, small y. So mark this. This is round, small y is green, and finally we get small r, small y. That is similar to the parent, wrinkled green. These are the 16 possible outputs of the F2 generation. When we do F1 into F1, 16 possible combinations are seen in the second filial generation. It is very simple. Now, if you look at the phenotypic ratio, how many round yellow, how many wrinkled green, and how many round green and how many wrinkled yellow. So if you count round yellows, 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 round yellow, which is similar to the parent. Parent was also round yellow. Then we have 1, 2, 3 round green. There are 3 round green. Round green combination is nowhere seen. So this is a new combination, round green. It was not seen in F1. It was not present in the parents also. Very important. Then we have another combination, wrinkled yellow. That is 3. 1, 2, 3. There are 3 wrinkled yellow. This combination is also nowhere seen. And finally we have 1 wrinkled green. It is seen in the parent. So the phenotypic ratio of a dihybrid cross is 9:3:3:1. These two new combinations that suddenly arises in the F2 generation, that is very important. Now, based on the production of these two new combinations, based on the dihybrid cross, Mendel proposed the third law, that is the law of independent assortment. Assortment is to segregate independently. What does this law say? It says that in a dihybrid cross, so in a hybrid that which containing two pairs of contrasting traits, contrasting traits. Example is what we discussed now. This is a hybrid. It contains two pairs of traits. So what happens is the segregation of one pair of trait is independent of the other pair. Is independent of the other pair. That is the law of independent assortment. What does this mean? It is very simple. This is round and yellow. Now, when these will get segregated during the gamete formation, that is RRYY, how do they get segregated? They segregated in such a way that the movement of one pair of alleles is independent of the other pair of alleles. So, when they get segregated, R can move with capital Y or it can move with small y. Similarly, the small r, it can move with capital Y or it can move with small y. So, if you look at the possibility, each possibility it is 25%, and this is total 50%, this combination, and this is 50%. So if it was not true, if the movement was not independent, then we will not get equal distribution of alleles. That is the law of independent assortment, and it is because of this independent movement we see the two new combinations, round green and wrinkled yellow, which was not at all seen. If the movement was not independent, like we expected in the monohybrid cross, in the F2 generation we got tall plant and dwarf plant, which is originally seen in the parents, whereas here we get two new combinations which are not at all seen in the parent or in the F1. Where did they come from? They come from the fact that alleles can move independent of each other. That is the law of independent assortment and the dihybrid cross. Let us say, try crossing a pure tall plant with, let us say, green seeds into a dwarf plant with yellow seeds. Check whether you get the same results in F1 and in F2 or not. So this you can use it as well assignment to understand whether you are able to form the Punnett square and get to the final result. The next topic is incomplete dominance. In the monohybrid cross, that when you cross a dominant plant with the recessive plant, in the F1 generation all plants will be dominant. They tried doing the same experiments on other plants. One of such plant is called as the dog flower or the snapdragon plant. The snapdragon plant belongs to the genus Antirrhinum species, Antirrhinum majoris. It is the genus. It is called as the dog flower, snapdragon. Now, in this plant, it produces two sets of flowers, red flowers and white flowers. So this is one pair of contrasting character. One type of plant produces red flowers which are dominant. Red flowers are dominant, whereas the white flowers are recessive. So the same crossing done by Mendel was followed over here. Plant containing red flower, red is dominant, so we use capital R. It was crossed with the pure white plant, don't put W. We have to use the small case of the same alphabet R. Don't make the mistake. So this is your parent. So red flowered plant was crossed with the white flowered plant, and we have to show the gametes. When gametes are formed, all the gametes will contain the R allele, whereas here in this case all the gametes will contain the recessive allele, and when they fertilize on fertilization, the F1 generation, capital R, small r, in the case of the snapdragon flower, the result was that the F1 generation plants had pink flowers. This is not in line with the observation of Mendel. In this case, it was neither red nor white, it was pink. So a intermediate character, in between intermediate character or trait was seen in the F1 generation. That is your incomplete dominance. Why does this happen? This is because we have a dominant allele and we have a recessive allele. In this heterozygous condition, hybrid condition, the dominant allele is not completely dominant, it is partially dominant. Therefore, it allows some expression by the recessive allele. Therefore, we see the pink color. And when F1 into F1 was done, when these plants were crossed, F1 into F1, that is capital R small r into capital R small r, when selfing was done, when they produced the gametes. So what are the possible combinations? This is one combination. The same gamete can fuse either with this, or with this. This we can do with the help of Punnett square also. See, capital R, we will get capital R small r, capital R small r, and small r. This is the F2 generation. This is the F2 generation. In the F2 generation, the plants were red, these two plants were pink, and this is your white plant. So the phenotypic ratio of a incompletely dominant plant is 1:2:1, and the genotypic ratio also is 1:2:1, whereas in the monohybrid cross the phenotypic ratio is 3:1. There were 3 tall and 1 dwarf, whereas here there is 25% dominant plant, 25% recessive plant, and 50% of the plants were of intermediate character, which is due to incomplete dominance. Why is a particular allele dominant, or why is a particular allele recessive? This is a very important question. Now you saw in the case of monohybrid cross and dihybrid cross in the F1 generation, always the dominant traits are seen. So in a hybrid condition, in a heterozygous condition, we know that there is one dominant allele, there is one recessive allele. Now in diploid organisms, there is a gene which is producing an enzyme. Therefore, the gene will have 2 copies of itself in 2 chromosomes. So there are 2 alleles of the same gene. The 2 alleles could be same, that is in a homozygous condition, or the 2 alleles could be different in a heterozygous condition. So take this example. So there is a gene that is responsible for the production of enzyme. Let us say the gene is E. It will have 2 copies. Both the copies or alleles could be same, or the alleles could be different. In this condition, when the gene coding for the enzyme alleles are different, what happens? There are three possibilities. The recessive allele. Why is it recessive? Why is it not able to produce the phenotype in the F1 generation? This could be due to mutation. There could be some alteration or changes in the DNA of this particular gene, due to which it is not able to express itself. Another important factor is the gene in itself is not responsible for the phenotype. Remember, the gene is going to produce a protein. This protein is responsible for the phenotype, the features. So, if the allele is modified, if there is a change in the allele, what could happen? There are three possibilities. It may produce the same enzyme here, means there are no changes at all. There are certain changes in the DNA, but with respect to the production of the product, that is the enzyme, it produces the same enzyme or maybe a less efficient enzyme, but it produces it. This could be one of the outputs, less efficient enzyme. In second case, it could produce a non-functional enzyme. Because of this change in the DNA, the enzyme could be non-functional, or because of this change there is no enzyme at all, and these enzymes could be responsible for the conversion of certain substrates. In these two cases, when the enzyme is not at all functional or when there is no enzyme, there is no conversion of substrates into products, means there is no phenotype. Phenotype is absolutely absent. Whereas in this case it is mostly seen. There is no effect because the phenotype is affected by the dominant allele. Whereas in these two conditions, the product is not formed, the phenotype is not at all seen. So that is why in a hybrid condition, the dominant trait is seen because the recessive allele is not able to produce the particular protein, or it is not producing it in enough quantity for it to show up. That is the concept of dominance and recessive. The next topic is codominance. As the word itself tells you, co means together, dominance. So let us understand this with the help of an example. The best example to understand codominance is the ABO type of blood group in human beings. We know that in human beings there are four types of blood groups: A, B, AB and O. Now these four blood groups are due to certain kinds of sugar polymers present on the membrane of the RBC. So in the blood we have RBC. The membrane of the RBCs, they have sugar polymers. These sugar polymers are under the control of a gene called as gene I. This gene I has three alleles. So the alleles are A, B and a small I. We know that usually one trait is controlled by two alleles because there are two copies of the chromosome. There are two alleles, whereas in this case of the human blood group there are three alleles. This is a best example for what is called as multiple allelism, where one trait, that is the trait here is the blood group, is controlled by more than two alleles. So this are examples for multiple alleles. The phenomenon is called as multiple allelism. Here the allele A is a dominant allele. The allele B is also dominant, whereas the allele I is recessive, meaning it does not produce any sugar polymers. It is recessive. So, how is this responsible for the blood groups? Let us write the possible genotypes and the phenotypes. So, in the case of this combination where both the chromosomes contain the same allele IA, both are dominant, the blood group, the phenotype is blood group is A. Also, if the genotype is IA and small I, it will result in A because I is recessive. So these two genotypes IA into I will result in the blood group A. If it is for blood group B, then you know it is IB, and another possible combination is IB into small I. This will result in the blood group B. And what if both the alleles are coming together, that is the A allele and the B allele? Do you get blood group A, or do you get blood group B, or do you get, like in incomplete dominance, the intermediate character? In this case, what happens is we get the blood group AB. This is due to co-dominance, meaning both the alleles are equally dominant. Therefore, both the alleles express themselves, resulting in the blood group AB. Whereas in the case of incomplete dominance, one allele was less dominant over the other. Therefore, the recessive allele was able to show just a little, and we get in between characters. Whereas here, both the alleles will express themselves in the heterozygous condition. Therefore, this is what is called as codominance, resulting in blood group AB, whereas blood group O is when both the alleles are recessive, that is I into I. So here you can very clearly see that the blood groups are controlled by more than two alleles. This is an excellent example for multiple allelism. And AB blood group is an example for co-dominance. In the hybrid condition, both the alleles are expressed equally. So that is all for this session. Thank you.